This video will focus on a historical perspective of heredity. So, what is heredity? When we say heredity, we're saying this is the transmission of traits from one generation to the next. Whereas genetics is going to be the scientific study of heredity. So, for example, let's say that we're looking at a mother and a father, and we say that mom has brown hair and dad has red hair. Heredity would be the passing of the hair color trait from the parents to the offspring. Whereas genetics would be looking at why does this child express brown hair instead of red hair like their dad. There's a little mystery behind heredity, historically speaking. Attempts to try to explain inheritance date way back all the way to the Greeks. They would say that there would be particles called pangenes that would travel from each part of the organism's body to the eggs or sperm and then are passed down to the next generation. But this is not quite right. Why? We know that changes in our somatic cells, so these will be our body cells, like our skin cells, for example, those do not influence egg or sperm traits. Another theory was called blending inheritance. In the early 19th century, biologists established offspring inheritance traits from both parents. Many scientists favored the idea that the offspring were kind of this perfect blend between the mother and the father, so like a 50-50 mixture between the two. We also know this isn't true. Why? Let's say that a black lab and a brown lab were to mate. They would produce a puppy offspring that had a very distinct color. So that lab, for example, may come out to be black or brown. We don't expect to see an offspring puppy that's like a perfect 50-50 mixture of black and brown lab. So therefore, this shows us that it's not a blend, right? There are distinctive traits associated with inheritance. Both of these hypotheses were later disproved by the scientist that goes by Gregor Mendel. Gregor Mendel is responsible for what we now term Mendelian inheritance. He is given credit for discovering what we call the mode of genetic inheritance. He replaced the idea of blending inheritance with this idea of particulate inheritance. He showed that characteristics can be passed down from generation to generation through what he termed discrete heritable factors. At that time, DNA hadn't been discovered. So Mendel didn't know what a gene was or an allele was, so he used this word discrete factor. We're passing down these distinctive factors from one generation to next, from parent to offspring. We now know what Mendel was describing is what we know as a gene or DNA. Mendel's theories from the 1860s have proven through our modern understanding of genes, chromosomes, and DNA characteristics. So, again, his theory was the strongest theory of all of the ones that we have talked about historically so far. And then we now couple that with our more modern understanding of DNA and genes, and we replace words like discrete heritable factor now with words like gene. Let's take a look at Mendel's experiments. Mendel did pretty simple experiments inside of a garden, and he focused historically, the most famous ones were on pea plants. What he was doing was studying different characters. A character is going to be a heritable feature that can be different from individual to individual. So let's say in these pea plants that he was studying, Maybe one particular character that he would look at would be flower color. So purple flower versus white flowers on these plants. For each character that had what we term true breeding lines, they could exhibit two alternate traits. So a trait is going to be a variation of a character. So let's say again that he was looking at flower color, then the traits would be either purple or white flowers. Purple and white are still a flower color, so they're talking about the same character, but we're saying that there are different traits associated with that character. He would then cross his pea plants and study these alternate traits. So, for example, let's say that he took one plant and the first trait would be purple flowering plants, and they could only produce purple flowering offspring. That's what we mean by true breeding. And then he would take the second trait of white flowering plants that could only produce white flowering offspring. What that's saying is the purple flowering plants in their gametes, so in the sperm or egg, they're only carrying the trait for purple. Whereas in the white flowering plant in the gametes, the sperm or egg, they're only carrying the trait for white. He would then follow these plants through 2 generations. He would look at the various proportions in the phenotypes that these offspring would have. He did this through looking at many different characters. So, for example, he did it with seed shape, seed color, pod color, flower position, and stem length. And what was interesting to Mendel is every time that he did this cross between these two different traits, he observed a very similar outcome in these individuals of about a 1-to-3 ratio in the offspring. Let's take a look at how he actually did these experiments. Okay. Mendel was very strategic and very methodological about how he performed these experiments. When you think about pea plants, so flowering plants, they make a flower and or a fruit. How did he make sure he knew like what plant was breeding with which plant? What he would do is he would take the first parent plant and he would remove the male reproductive structures called the anthers right here. The anthers are responsible for producing sperm. Now, flowering plants can have both male and female reproductive structures on them. So he wanted to make sure that he removed those. And then he strategically did the pollination. So he would go to an opposite plant, such as a white flowering plant, and then he would use a brush to very gently take the pollen from this white flowering plant. And then he would bring that pollen over to the female reproductive structure on the opposite purple flowering plant. And then he would take a look at what were the progeny or what were the offspring that they would produce. There are a few important definitions that we should talk about. It's not that I'm going to sit here and quiz you per se on each definition, but we will be using these definitions when we work example problems. When we use the word genetic cross or hybridization, we're saying that we're breeding two organisms that have different traits. A hybrid is going to be the offspring produced by these two plants. Then we have what's called the P generation, the F1, and the F2 generation. The P generation, also known as the parent generation, is what we start with. So those initial true breeding parents. The F1 generation is going to be the offspring that are produced from those parents. The F2 generation are going to be the offspring of the F1. So, for example, the F1, if we're talking about flowering plants, it could self fertilize or You can take two F1 generations and breed them together to get to the F2.